Okay, so if cancer can be thought of in a basic sense as our DNA getting messed up through mutations, where do these mutations come from? I think it's very easy to think, oh, mutations come from carcinogens or exposures to different things from the outside. But that's only part of the story. If you consider that there are three main ways that our DNA is under threat of mutation, exogenous mutagenic factors like carcinogens or UV radiation, that's only one of the three. There are also, simply put, different possibilities of our DNA being damaged because of just normal biochemical processes inside of the cell, having to do with, for example, metabolism. Or even more fundamentally, just the very fact that in order for our cells to replicate, we have to make copies of our genome to be able to make two daughter cells. Mistakes that happen during DNA replication in S phase is a very real threat of actually getting mutations that start a cell transforming. So to wrap up today's lecture, let's just look at this first part, and then we'll continue on the next time with the other two. So I think you're all aware that DNA being copied during S phase is copied by DNA polymerase complexes. So in this slide here, we have a complex of enzymes that include DNA polymerase and different types of functional units of the polymerase holoenzyme that all work together as a large machine that you can see is writing down the DNA. So it's copying the template strand here, and it's making this brand new copy of the strand. So as it's moving along, of course, it's pulling or incorporating dNTPs, A's, T's, C's, and G's from its surroundings and adding it to the next spot where it's supposed to be. So this process is not 100% foolproof. Every so often, the DNA polymerase complex will make a mistake and add the wrong nucleotide. And so it has proofreading. Oh, so by the way, a lot of times these cartoons, I find them very whimsical looking because I always wonder why the illustrator chose this particular design. I kind of like it because it kind of reminds me of, like the old Beatles' Yellow Submarine. But anyhow, so here what you can see is that there are different components of the complex that actually can recognize that a mistake has been added. And so in this case, these different components recognize that there's this weird little bulge in the DNA, and so the whole complex can back up and repair that mismatch before then continuing on. And that proofreading process is very effective. Another way that the DNA mistakes in DNA replication can be repaired is, let's say, for example, on the top here, that whole DNA polymerase complex has moved on, and it unfortunately missed this mismatch right here. So there are enzymes that are essentially coming and docking on the DNA and helping to catch these types of mistakes after the fact. These DNA enzymes, these mismatch repair enzymes, or MMR enzymes, include proteins such as MutS and MutL. So these were originally named or identified in bacteria. So these MutS, MutL proteins, they're constantly scanning the DNA. And if they encounter something like a little bulge where you have a mismatch between the new copy of the DNA and the original template nucleotide, they can go ahead and actually cause a section of that mismatched DNA to be excised out, so trimmed out, like such here, and then DNA polymerase can come back and fill it in. So how is it that these MutS and MutL actually detect a mismatch? They do it by riding along the DNA. So here we have that dimer of MutS. These are the ribbon parts of it. They have different domains to them. And you can see that these are only parts of the protein that are shown here. right? So it's not like the protein is actually three separate little parts that are working together. The whole protein would be kind of wrapped around the DNA, but for clarity's sake, they're only showing the parts or the regions that contact the double helix of the DNA. So this protein wraps around the DNA and it basically rides the DNA, just kind of like a roller coaster, spiraling around on the alpha helix. And just as long as the copy strand that was made has a nucleotide that matches the template strand, you're going to have a nice alpha helix for this enzyme to ride on. But if you have a spot like this spot here, where the copy nucleotide that was put in is not the correct match or the complement to what it's supposed to be, the alpha helical structure of the DNA at this point is going to be bent a little bit. So as the MUT-S enzyme is riding along, spinning sort of along the spiral, it gets to this bend and it gets stuck. It literally gets stuck in this corner because it can't work its way through. And when it gets stuck in this corner, its shape will change because the shape of the protein will get a bit deformed because it's stuck in this little bend right now. And that will reveal different parts of the protein that then attract different types of enzymes that can go ahead and now cleave the DNA as part of then fixing or remaking this section of the DNA strand. Okay. So I mentioned that overall DNA repair during replication in S phase is very efficient, right? That's not surprising, because if it weren't, then every time our cells divided, we'd always have lots of problems. So the DNase polymerase itself, the complex, as it's going along and adding new bases, it will make an error in about 1 in every 10 to the 5 bases. Okay. Then if it can back up and it does its 3' to 5' proofreading, most of the time it'll catch the mistake, but even that 3' to 5' proofreading results or leaves behind mistakes in about 1 in 10 to the 2. So about 1 in every 1,000 mistakes, nucleotides, it makes a mistake. Then lastly, the MMR enzymes like the MUT-S, they will come along after the fact and scan the DNA. And they usually do a really good job repairing, but they allow maybe 1 in every 100 bases to slip through. So you might say, well, that doesn't seem very efficient, 1 in 100 bases, right? But keep in mind, that's only after you've already had other mechanisms that are much more stringent in catching mistakes, 1 in 1,000 or 1 in 10 to the 5. So in reality, the overall miss rate then of allowing a mismatched nucleotide to be part of the genome is about 1 in 10 to the 9, or about 1 in a billion bases. So that sounds pretty good, but then you still have to think about the fact of how many base pairs the entire genome consists of, right? Because the human genome has about 3.2 billion base pairs. So that means with every cell replication that takes place, your genome is going to get 3 new mutations in those daughter cells that weren't in the original cell that divided. Right? So if you think about the sheer number of cells in your body that are dividing every single day, what does that mean? That means that because we have about 37 trillion cells, your genome every single day ends up getting thousands of new mutations just from the process of mitosis alone. And so I think that's a really kind of eye-opening thought to think about, that we are constantly, every single day of our lives, incurring thousands of new mutations in the genomes of our cells overall in our body, simply as a result of our cells dividing.